Welcome to day 3 of the Phage Annotation Workshop, where I'll give you a short introduction to classification and taxonomy of phages. So the aim of today is not to become an expert in phage taxonomy. The aim is for me to provide you with the information and tools you need to fill in the organism section, so this section of a GenBank file. And one of the things that I'm showing here is that this is one of the first phages that I've ever isolated in sequence, and you see that it's already 10 years ago that I submitted this. And when I submitted this Pantoja phage limelight, it was classified viruses, double-stranded DNA viruses, no RNA stage, Caudiviralis, Potoviridae, Autographiviridae, unclassified 5K and V-like phages. And we even mentioned that 5K and V-like viruses in our publication. Now, a lot of time has passed and the taxonomy has been updated, and you can see in my GenBank file that the date is still the original date that I submitted. But in the meantime, this has now become viruses, diplo-DNA viria, Hungong virae, Uroviricota, Caudivirices, Caudiviralis, Autographiviridae, genus limelight virus. So that means that in the background, the taxonomy has been updated and you don't have to do anything for it. So the idea is for you to, at the time of your submission, provide the most updated lineage information as possible, and then add, unclassified to the highest or the lowest taxon that you can find. One of the important things is to do not use taxonomy information in the phage name. I've seen recently quite a lot of bacteriophages. Some of them, or maybe most of them, derived from metagenomic sequences where they write something like Myoviridae species something something, and that's the definition and that's the source. And then, in a couple of years, when something gets updated, that name will still be Myoviridae species something something, but Myoviridae will no longer exist. So just don't make it more complicated than it is. Provide your phage name as the organism in the beginning, as the source, and in the definition line. This always happens when I do something. Okay. These are some resources for that recently myself and others have provided, some communications or opinion pieces on naming, classifying, and the route forward for genome-based phage taxonomy. So we'll start with naming. It might seem, pedestrian, but this is quite important because there are no official rules about naming phages or virus isolates from the ICTV standpoint, so the International Committee on Taxonomy of Viruses, but there are a lot of rules for the official taxon names. And because we, as the ICTV Bacterial Viruses Subcommittee, have used phage names as basis or inspiration for some of our species and genus names, we cannot always 100% match these because we have rules, whereas you don't. But the most important thing is to be unique. And remember, a phage species is not the same as a phage. The same way that all domestic dogs belong to the same species, but they are not the same. So this is a very important distinction, meaning that there can be multiple phages that belong to the same species. We have, as the ICTV, recently implemented a binomial species naming system, and all current names are being transformed from their original name that could be kind of in any format, so the species name, into a binomial format. So for example, Salmonella phage P22, which is a member of the genus Lederberg virus is now the exemplar isolate of the species Lederberg virus B22. So this is a free-form binomial format for the species name. And the exemplar isolate of that species is Salmonella phage B22. Same goes for Enterobacteria-phage MS2. It is a member of the new genus MS virus, and it's an exemplar isolate of the species MS virus zinderi. And in this case, the people who submitted this taxonomy proposal have chosen to take a Latinized binomial approach. Our hope was that this would make a clear difference between the phage isolate and the phage species. So we would not want you to see that people suddenly start naming their phages according to a binomial system, because then there will be chaos. In practice, you'd only need to mention this once at the beginning of your paper, saying my phage is called Salmonella phage Tweedledum, and it belongs to the species Lederberg virus B22. And for the rest of your paper, you just talk about Tweedledum. Okay, that being said, I wanted to get that out of the way because it's quite important. Let's get to the crux of it. What is taxonomy? Not to be confused with taxidermy, the practice and science of classification of things, concepts. It is written often by taxonomists for taxonomists, and it's so dull that few, if any, non-taxonomists are tempted to read it. Yes, maybe. Most subjective branch of any biological discipline, in many ways more of an art than a science, possibly. I also like this quote, that taxonomists like to think of ourselves as artists able to perceive form, shape, color, and relationships that are hidden from the gaze of the more mundane scientists. Just to say that some taxonomists are quite full of themselves. In phage taxonomy, one of the first papers that I've ever read about this was we need to agree to disagree. Hopefully, we've kind of moved on a bit from this, but the last one I wanted to share was this, because it starts with, what do you think of when you think of taxonomy, an 18th century gentleman in breeches? Well, I hope I am not perceived as that, and that we can bring a little bit more pizzazz to taxonomy. That being said, it's actually all about putting things in boxes. So classification is putting things in boxes, and then when you come to taxonomy, it's the science behind it, but it also includes nomenclature. And currently for viruses, we have 15 ranks or 15 boxes in which we can classify, from realm down to species, which is the most narrow definition. Anything below that we don't do. That's not in the remit of the taxonomists, that's in the remit of expert groups all around the world who are expert in a particular type of virus. We have to follow nomenclature rules, and we have to follow certain rules on how to write things, or orthography rules. Practically, how does taxonomy work? Virus taxonomy work, so that's also for phages. It's done by committee, not by priority of publication, meaning that we do taxonomy by submitting taxonomy proposals, which can be written by any member of the public, which then get submitted to the chair of the subcommittees. We have a bunch of subcommittees here. So I am the chair of the bacterial viruses subcommittee, meaning all phage proposals come to me first. And then there's a whole cycle of approval. And at the end of that cycle, the taxonomy is updated. So that is not by priority of publication, meaning that there can be differences between the first person who describes something and comes up with a system to classify and name it. If that's not consistent with what the rest of the ICTV is doing, it might not get accepted like that. What's the process? So people, whether they're members of the ICTV or whether they're members of the public, submit their taxonomy proposal to the subcommittee chair. Then the the subcommittee chair compiles everything, maybe provides some feedback to update it. And then all of these proposals get submitted to the executive committee. The executive committee reads everything and then approves proposals or rejects or suggests corrections. Then for those that were corrected, there's another email approval vote. That's usually September, October, or November in the case of this year. Then there's a ratification vote of all of those approved proposals by the membership of the ICTV. And when that's approved, the taxonomy is updated, and then we publish the master species list, an MSL, and the virus metadata resource, which is another Excel file, which links taxonomy to accession numbers in NCBI. So at all stages, these proposals are available from the ICTV website, and at all stages, proposals can be withdrawn or deferred to the next year. So the best case scenario, when you find a new virus and you find a new phage and you get it really quickly sequenced and you do your analyses and you submit your proposal for the May deadline, in best case scenario, in March of the year after, it is officialized and then will get updated in the taxonomy database as well as the NCBI taxonomy database. So what does that mean for all the viruses? And in this case, the bacteriophages. If I'm looking at the number of nucleotide phage records in NCBI over the years, I chose this year because this is when I started getting involved in phage research. And we can see we were well below 10,000. So these are all records, including incomplete. So if it's just, you know, a primer sequence or an amplicon sequence, that's also in here. And we've gone rapidly up. At the same time, we have the RefSeq phage records, which are the ones where the accession number starts with NC underscore. Those are now only about 4,000 odd something, or maybe 5,000. And that is because this is a subset of the phage records. And since I think 2019, we started working together with the NCBI so that the phages that are in those records are the ones that are the exemplars of species from the ICTV. Meaning that if you have a species containing a lot of phage isolates, where the phage isolates are very similar to each other, and there's, for example, 100 deposited in the database, of those 100, only one, which is the exemplar, will be in the RefSeq database. If we then compare to a metagenomic database, like the IMGVR, you can see that at version 3, it was at about 3 million sequences and about 1 million OTUs, which are roughly associated with species. And then you can see that the majority of current, you know, phage records are unclassified. But not to despair, I told you about the 15 rank structure, starting at the realm, at the top or the bottom of the pyramid, depending on how you organize it. These are all the available hierarchical ranks that we can use for classification. If you, for example, look at this one, this rank, the class, we now have the class Caudoviruses. And whereas, you know, the majority of sequences are unclassified because we haven't defined a species and a genus for them, that doesn't mean they don't have a place to live. All of the tailed phages can be placed here in the class Caudoviruses. So that is not too bad. So at least you have something to fill in the organism field in your GenBank record. So how does your basic phage classification workflow work if you have a new phage? So you start with your well-annotated phage genome, which hopefully you know by now how to do. The first thing to do is find your database relatives. I always start with a BLAST-N type search, because if your phage is very similar to anything in the database, that makes it really easy to find its location within the taxonomic tree. One of the other things that we'll show today is how to use VipTree, which is based on the phage proteomic tree underlying algorithm. But there's other methods to use to find database relatives, depending on how similar your phage is to phages in the database. First thing then to do is, if you have any genomic relatives, is to find the intergenomic distance. We will show how to do that with Veridic today. Then find your shared protein content, and then basically do a multiple sequence alignment and phylogenetics. And this can be of signature, it can be of one signature gene, multiple signature genes, or it can be of a set of signature genes or core genes together. And basically, when you report it, should you submit a taxonomy proposal, it's best to kind of use all of the information collected along the way. Step one will be, how do you find your relatives and how closely related are they? If you do a BLAST search, you can see this, basic BLAST search query. I suggest you limit your search to a subset of organisms. And you can do that here in the organism setting. So when you start typing here, there's options that will pop up. You can, if you are absolutely unsure what type of phage it is, you can limit your search to viruses. If you know it's a tailed phage, you can limit your search to Caudovirales or Caudovirusites. If you have a prophage, then you could also limit yourself to bacteria, but I would do the search twice then, once for viruses and once for bacteria, because you might just see that your prophage matches this region in all of these bacterial genomes, but it will not give you any information on taxonomy. So that's BLAST-N. BLAST-X, you can compare your genome to a protein database, and that's if your BLAST-N doesn't yield a result, meaning that there's nothing in the database that is sufficiently similar to your genome. You could also do a T-BLAST-X, which compares your genome as a translated query to a translated genome database, but it's very computational demanding because it will check all of the 6 frames. So I would not recommend doing that with your full genome. You could cut your genome into pieces, or you could try and do it from the command line. But for this, we actually do have VipTree that we can use. So there's an alternative location to start your BLAST, and that's NCBI Virus. And I've recently started using it much more because it's a very nice feature, a very nice resource available from NCBI specifically for virologists and also phage biologists. So this, when you search here, you can search by sequence or by virus name. And it's automatically limited to anything in the virus database. It has an easy refinement of search results. It has an extensive metadata table, and you can do a more detailed investigation of your search results by clicking on certain of the options, and it also has an easy download function. So I do suggest you look at this, and you can click through all of the records that are available. Should none of these kind of BLAST searches find anything or give you a good enough result, you can try the phage proteomic tree or the VipTree. So this is an online resource where you can upload your genome, up to 100 genomes. And this basically makes a comparison between 2 sets of genomes pairwise across all of the RefSeq database in this case, this is for double-stranded DNA prokaryotic viruses. And it compares, so the predicted proteomes using T-BLAST-X for every combination of two genomes. So, and it will report that in this kind of tree. And the first thing that you can see here, this is just a reference tree that's available if you go to the main Victory page without having uploaded anything. And what you can see is all of these branch lengths being really long. And you can see the scale here. It goes from 0 in the middle to 0.5 at the ends, meaning that between two genomes, the maximum length is 1. And these branch lengths are scaled so that at 0, so if two genomes share, have a branch length of 0 or 0.0 on this tree, that means that they are identical at the amino acid level. And if your branch goes all the way to the middle, so it's 0.5 times 2, so 1, then they share nothing. So all of these large branch lengths show you that within these double-stranded DNA phages, there's a large number of groups of phages that share nothing at the amino acid level. And this is why this can be a good feature for you to find your relatives, because if you are able to place it in one of the branches where something like this, where you have a clustering, then you know that these are your closest relatives. And then you can start working from here. If you're unfortunate and your genome has a branch length that is just 1 with everybody, then, well, you're either unfortunate or very fortunate because you found a really novel phage. And just to say, the taxonomy of this inner ring is not up to date because we have many more phage families at the moment. So now we've found a group of, hopefully, a group of relatives, and it's time to determine their intergenomic distance. And this can tell us whether your phage represents a new species. And we're using Veridic for that. And this is a tool that looks at, pairwise looks at how related to phages are at the nucleotide level, and our current species demarcation criterion for bacteriophages is that they need to share at least 95% of their genome over the whole genome length, meaning that they can, genomes of two isolates belonging to the same species differ from each other by less than 5%. One of the important things is to check for synteny of the phage genomes, and I will show you why. And that's this one here. You can see that basically here on the top right-hand side of our Veridic plot, where 2 by 2 each genome is compared, you can see that they all share 100% identity. But as a test set, what was added here was the same genome inverted and permuted. And this, all of these instances show you the same number in intergenomic similarity, but there's differences in the genome rearrangement. So it's always good to look at your genomes as well. So this is for the species level. You can also use this program for the genus level. And then the cutoff is 70%. And that's basically what you can see here, that this is a group of phages that belong together at the genus level, with this one related, but an outlier. And this is how you can use your intergenomic similarity or nucleotide identities to start looking at the species and the genus level. Now, for the genus level, what we also need is that it needs to be a cohesive group of viruses that also is monophyletic in its marker genes. Why do we need this? Because the ICTV, as a rule, needs phylogenetic analysis to back up its its taxonomic proposals. Other potential defining characteristics that can be there is your average genome length, number of coding sequences, your percentage of shared coding sequences, the genome organization, all these type of things. If you think you have the first representative of a new genus, contact us about this. So this is the intergenomic distance. Then we get to the shared protein content. And then we are in the realm of, does my phage belong to an existing subfamily and family? So this is assessed with a combination of genomic, proteomic, and phylogenetic tools. And one of the things that I can suggest to you is we have recently defined a whole number of new families of tailed phages. And check the taxonomy website and check the history tab here, where you can see all of the proposals. And then you can check for each of these in the proposals what the criteria were to define these new families, so that you can easily know what marker gene to use if it's similar to yours. For example, members of the autographic viridae have an RNA polymerase, but members of other families may not have an RNA polymerase. So an RNA polymerase would therefore not be a good marker gene for any of the other families, but it is a good marker gene to use for the autographic viridae. If your phage is so new that you need to start defining new subfamilies and families, then we're getting into the realm of advanced taxonomy, and I do suggest you start contacting members of our subcommittee. If it falls into some of the new families, you can have a look at some of the papers we published on each of the new families, or the taxonomy proposals, or the ICTV report chapters. If you think, okay, we're in the realm of we have a new family, you'll have to start defining the core genome, meaning the genes that are shared across all members of the family. There's a web server that you can use to do that, and there's a bunch of command line tools which have been mainly developed for bacterial pangenomics. But again, this is in the realm of advanced classification and not in the scope of this workshop. If this is the case and you want to fill in your GenBank form organism line, then you just have to fill in unclassified caudavirus seeds and be done with it. Because what we're currently in the process of doing is abolishing the families Myoviridae, Podoviridae, and Cypheviridae, and the order Caudoviralis. So all tailed phages will be moved one level up, which is the class Caudoviridae, and the morphology-based families Myoviridae, Podoviridae, Cypheviridae will be removed so that we have space to get more genome-based families. And in the meantime, there will be a bunch of genera that are directly within the order of within the class Caudoviridae. So there should have been class, there should have been class. So directly within genera, subfamilies, families directly within the class Caudoviridae. And this is the case because we don't want to define new families without having enough information and enough members. So if we have a new phage, which is so new that it would be the first member of a new family, we will probably not define a family in the beginning, because then we don't know what the demarcation criteria will be. So finally, we come to phylogenetics for phages, and then often the question is what marker gene to choose. If the best practice is to choose more than one, and if you are familiar with any of the core gene methods, then you can even do phylogenomics, meaning you can do a kind of a concatenated marker gene tree for all of your core genes, but I will not elaborate on that today. So one of the best things to do is to check the literature and ICTV documentation for the most appropriate gene for your group of phages. Try to include any outliers. And the process is relatively straightforward. Make a database with your marker gene amino acid sequences, perform a multiple sequence alignment, check it for mistakes, optionally trim part of the sequence alignment, and then construct a phylogenetic tree and visualize. I wanted to share with you a couple of examples of new families where we used the marker gene approach for phylogenetics. In this case, this is the new family Chaseviridae, for which the ICTV report led by Hani has just been published. And in this case, we chose the RNA polymerase and the terminase large subunit together, and we made a tree for both of these together. And it's a very straightforward family to make because there's not that many members yet. It can get quite complicated. So this is two new families of T4-like phages that were proposed, and this taxonomy proposal by Andy Millard and colleagues is currently in submission and has been approved by the ICTV and will be voted on early in 2022. And in this case, the tree was built on a combination of 14 core genes combined to really get the resolution necessary to get a lot of members together in a tree and to get the groupings to be robust. In summary, to classify your phage, you need to find your relatives in a public database, identify your relationships at the nucleotide level, identify relationships at the predicted proteome level, perform phylogenetics, fill in your information in your GenBank file, and if you are willing to, if there's something new, submit a taxonomy proposal to a study group chair or subcommittee chair. Now, one of the things we do say to people who want to submit a taxonomy proposal for new taxa is, it's not enough to just have your own phage and submit the new proposal. You need to have defined the new taxon with all of its members, meaning you also need to include the database relatives and get all of that information as well. It's not enough to just say, I have one phage, it's super similar to all of these, but we're not including these in the taxonomy proposal. And with that, I'd like to thank you all for listening. Thanks, Andrea. Thank you, Andrew, Hani and Dan, and everybody who works with me.